Hello everyone, my name is Ashton. I am a School of Plant Sciences and Ecology master's student, and today I'll be presenting to you about microbes in middle schoolers. So before I begin, I just kind of want to lay out the outline of what I'll be talking about. So as we were just introduced to, I'm going to give a brief overview of the SBAR project, which has brought me here today to present for you guys. Then I'll follow by talking about microbes, and then I'll be ending on the mysterious middle schoolers at the end. So first, let's go ahead and dive into our SBAR. SBAR stands for the Sustainable Bioeconomy for Arid Regions. And everything in this talk will be in the context of SBAR. So just keep that in mind as I move forward throughout these slides. So SBAR in itself is a small part of a very large project that spans the entire United States. As we can see from our map here, SBAR represents the Arizona and New Mexico section, while there are multiple locations across the United States that's looking at a similar sort of production. And just to note that within our SBAR here, we're looking at guar and mayuli plants, which I'll explain a little bit more later on. The ultimate goal of SBAR is to develop a system for feedstock production, or feedstock development, production, and delivery in the Southwest United States. And the part that I'm involved with is the development of education, in which for extension outreach to teachers, 4-H youth groups, and to farmers. So that kind of gives you where and what we do, but what exactly does SBAR do? The 4 bullets that I'll say are that SBAR, the idea of SBAR is to grow sustainable crops in the Southwest using native plants. Additionally, to identify new bioproducts from guar and mayuli. These plants have been used for a variety of different things over the past, and now we're looking at them as new sources for rubber, biofuels, and much more. And then our last bullet point is to develop a full assembly line from farmer who's planting the seed all the way to consumer who's using the rubber for a tire on their car. So this is a very large project that spans pretty much every discipline area that you can imagine. And to address these 4 points, our mission is ultimately, or our project mission, is to build a sustainable bioeconomy for arid regions to improve the quality of life in rural communities and native nations. And in order to complete this mission, or to achieve this mission, we are engaging growers, producers, and different industries along this production pipeline. We are also training the next generation of scientists, agricultural producers, and researchers through optimizing the production of guar and mayuli into new crops. And this is something that I want to note is really special. There are not a lot of crops that are actually grown in the United States. There's a very limited number. So the introduction of a new crop into the general pipeline of crops is a pretty big deal. And the crop that I'll be talking about today is mayuli in particular. I did also introduce guar, however, for the Arizona team, which is where I'm at, we're focusing mainly on the mayuli part, which the scientific name is Parthenium argentata. And in a map on our lower right-hand side of the slide, it can grow, or it's from locations such as California, Arizona, New Mexico, and as far east as Texas, and as south as Mexico. So it has a pretty large growing range, but we do see that there's a large overlap with our nice Arizona state. And what's really awesome about this plant is that it produces rubber and resin in the branches and roots. It's resistant to drought, which makes it ideal for locations such as Arizona. And all parts of the plant can be processed, meaning you can take the whole plant out of the ground and put it into a production pipeline. And then a special note for those who are interested in herbicide and insecticides, these are only needed for the initial seedling establishment. After that period, because it is a native plant, you don't need to apply any more herbicides or insecticides, making it an economically stable option. So what I just talked about, these are the key 6 concepts for the SBAR project. And the things that I just spoke about are predominantly this last section, or the bottom three, which involve the feedstock development and production for the Waiuli growth, post-harvest logistics and the co-product, so what can we take from those plants, and then system performance and sustainability, trying to do this process as sustainably as possible. So just to highlight this section. However, I'm not in that part. I am in the education section. So the work that I do is within education, which tends to overlap a lot with the extension and outreach, and the development of resources from this project to be available for the public. And you're probably thinking, why is he in education? What does he study that makes him so interesting to do this part of the project? So I'm going to give a little bit of background on my own research, which is involving microbes. And I know I just talked about plants, and now I'm talking about microbes. Specifically, I'll be talking about plant microbes. And on our bottom, we have a great photo of some fungi growing. So specifically, I look at microbes in the Southwest. And here's a photo from the Santa Rita Experimental Range. There's a nice cloudy overcast and it's great. My project is focused in this concept and idea of restoration ecology, in which we're looking at the recovery of degraded, damaged, or destroyed ecosystems. And this is becoming more and more relevant in our society today, as we're seeing that there is increasing levels of human disturbance in our environments, both at a local and global scale. Some projects to combat this disturbance include like the mangrove restoration project along southern Florida, the reforestation of deforested forests in the tropics, and also one that's a little closer to what we're talking about today is the revitalization of land that had been turned into farmland and then left vacant. So all of this is to say, we're putting plants in, we're attempting to reestablish these ecosystems by placing plants in these different locations, and there's a need to understand these factors that are influencing our plant establishment. So what can we do to help these plants grow faster, stronger, bigger, better, all and such? And that's where I come in, where I'm looking at microbes and seeds. Seeds are important for plant establishment, succession, and environmental resilience. We've all planted some seeds. We know that you need to get a plant. Soil microbes may influence our seed survival, germination, and the ability of seedlings to establish. And those are two important concepts when thinking that humans are negatively influencing our soils. So it's making our soils harder and harder for these plants to establish in these different environments. And more specific, I focus on campgrounds, which sounds a little strange, but campgrounds, if you think about it, you're going into the desert, you find a nice campground. It's a very centralized location of highly disturbed soil that's consistently used throughout the year. So it's perfect for looking at microbes in disturbed soils. So the research question that I had asked regarding this was, how does land disturbance due to camping influence the microbial colonization of restoration seeds? So basically I'm asking, in these disturbed soils, are microbes infecting seeds? And how is that affecting the seeds later on? And I'm going to skip all of the methods and just give you my conclusions. But I did make a really pretty picture that I wanted to share. So through this project, I had done microbial characterizations using morphology and sequencing. And here we have just a representative example of 16 of the different fungi or bacteria that had grown out of my seeds. So some of the takeaways that I got from this project thus far is that land disturbance is in fact affecting microbial colonization. When comparing our disturbed sites to natural sites, we're seeing different communities. We're seeing lower levels of fungi in one in comparison to the other. And we're also seeing that soil microbes may be associated with germination success, meaning that if there is a microbe or a fungi or bacteria that is associated with that seed, it may grow stronger or faster. And all of that is within this context of the SBAR theme of sustainable agriculture, which might sound like a stretch. It's not, I promise. We're trying to come up with a way to revitalize our ecosystem, and what SBAR is trying to do is a way to grow plants in our ecosystem with the least amount of disturbance to our naturally great ecosystem. So ultimately, it's the same sort of same sort of goal. So with that, and through my research, I've had the opportunity to work with a lot of outreach projects. So basically translating my experiments into curriculum and classroom units for teachers at the high school level specifically to use to help their students understand more about their native ecosystems. And with that, that's kind of brought me towards my interest in education as a total, and more specifically, pushed me towards an interest in middle school education. I know I said high school, but middle schoolers are pretty awesome. So, the way that I do that through the SBAR project is as a university fellow, and here we have our fellows and teachers from the 2018-2019 cohort. And what's interesting is teachers and fellows, such as myself, who are graduate students, are partnered together to work on developing SBAR education from the project and taking it into the classroom. And through this partnership, we're collaborating on lesson development. And this can take place, we have classes from 6th grade up to the high school level. And here we have myself with my teacher, Jamie Camaro from the Flowing Wells School District. What the fellows do in this partnership is we act as translational experts, translating the hard science from the experiment and the project to an understandable level for classroom curriculum involvement. We also act as the STEM field experience. Pretty much all of the fellows have some form of experience in a science, so we act as a resource for the teachers to pull from to answer any questions that they may have about the science. From the teacher's point of view, the teachers are working on curriculum design and learning expertise. And they are the learning expertise in the classroom, as I have not been in a classroom beforehand. And they're also helping us to understand the community implications of the SBAR work that we're doing. So as I had said, I was part of the 2018-2019 cohort. Here's another great group photo of all of us together. The teachers and educators range from middle school to high school, and our university fellows come from either the University of Arizona or New Mexico State University. As we saw, the SBAR project ranged over both Arizona and New Mexico. And our partnership all began in the summer of 2018. So during our first interactions as fellows and teachers, we were part of a professional development which took place over 2 weeks over the summer. In that first week of professional development, we can see here that we have a collaboration of everyone involved in this program. Here we have students, teachers, and fellows all working together to learn from researchers and extension experts from the SBAR project of the different activities and lesson plans that have already been developed. What's interesting and really powerful about this is everyone is on equal footing for this, as we're participating in lab activities the same as the 6th, 7th, and 8th graders, and we're learning it at the same time and thinking of ways of implementing this knowledge and information into the classroom. And from this first week, teachers learned and were able to start designing activities and lessons based on the SBAR curriculum they've experienced through this first week. And I like this photo at the very bottom on the left-hand side because we also got to go on a field trip or two, and it was really nice to get out there and experience nature with the eyes of a 6th, 7th, and 8th grader. So then the second week, the students had finished within the first week, and the second week focused on the partnership with teachers and fellows together. So here we have myself. I know a lot of these photos are me. I'm sorry, I just like them. So we have myself and my teacher, Jamie Camaro, and we are working on the curriculum map, which you can see in the center of our slide with the bubbles and the arrows and the confusion. We were working on the curriculum map for the entire year for 6th grade for her class. And what what happens during this time is we're bonding, we're getting to know each other, and we're also generating lesson ideas and implementing the SBAR curriculum through every single part of our curriculum throughout the year. And that's okay. And some of the roles of a fellow include weekly meetings, 10 hours dedicated to these partnerships, of which 8 are about, for me, 8 are spent in the classroom, developing lesson plans and helping in the classroom, and this includes demonstrations, grading, guest speakers, and more. And on here we have an example of seed bombs, which some of us may know are a restoration technique that many people use. And we also have a photo here from the Lost Carnivores Community Partnership, which is interesting because it's been an opportunity for myself to network with a lot of community partners that involve middle school education. So we have some such as the Lost Carnivores, Project Wet, the Sea Life Aquarium in Arizona, different departments in the University of Arizona, and also TEP, and even revisiting my own School of Plant Sciences and learning much more about the program than I had known beforehand. One form of lesson development that we've done so far was I invited a guest speaker, Liz Bowman. She is a PhD student in plant pathology who talked to us about climate change, native plants, and fungi and how they all interact with one another. Through this, one example of the work that we're taking away from this is the development of a PowerPoint, which then can be shared with other teachers, hands-on activities for students using native plants, and then ultimately the development of lesson plans that can be shared as a resource for other teachers nationwide. Soon, I'll also have another guest speaker, Desiree Kitzel, a master's student from plant science, coming in to talk about the native plant microbiome in the classroom. We have white lily plants growing in our classroom, thanks to the SBAR project, and this is coming soon with details. She'll be there in the next month or so. So in conclusion, SBAR is a multidisciplinary project. Curriculum is being developed for every stage of the production. Early academic outreach is a chance to engage many people. As we can see from my slides, it's not just myself who's being involved, it's different projects, different schools, different people. And ultimately, one takeaway that I want everyone to go away with is that native plants are important for our environment, so I would like you to start planting some. And with that, I would like to thank everyone who's part of the SBAR team, everyone who's a part of the Arnold Lab, and all of our funding sources for this project. And with that, thank you, and SBAR is now recruiting graduate fellows for next year if you're interested.